Hello, everyone, and welcome to today's webinar titled Utilizing Molecular Technologies to Drive Productivity in the Microbiology Lab. This panel presentation is a part of the 9th annual event in the Microbiology Week virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about Thermo Fisher Scientific, go to www.thermofisher.com. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Now let's get started. I now present today's speaker, Scott Brady, CEO and founder of Wise Diagnostic Systems. For a complete biography on our speaker, the presenter tab is located on the left of your screen. Scott, you may now begin your presentation.
Good morning, and thank you for that introduction. So my goal here is to talk to audience here about the operations and business side of PCR infectious disease testing. And I have the subtitle down there, just some things to consider and some strategies you might want to adopt if you're thinking about building PCR infectious disease testing into your laboratory. And I think this can help you whether you don't have any clinical testing right now and you're thinking of adding molecular testing specifically for infectious disease testing, or even if you have a clinical lab and you're currently doing culture and sensitivity and you're trying to weigh the pros and cons of replacing or adding this type of testing as a complement to the current testing that you offer. And so first, I'd like to kind of go a little bit through my background, and I think this is a little bit important to the side of the PCR testing that I'm trying to gear my presentation towards. And you'll see from the photos and just from the bullets that I did go to West Point. I actually studied undergraduate law and systems engineering while I was there, and I was then served in the combat arms and the field artillery for 5 years. And in fact, at the bottom here, you'll see my last job here. I was an instructor, and that down there that looks like an old school slide rule is actually what we call a graphical firing table. I was teaching young lieutenants how to fire artillery, miles and miles away and hit a target on that. And then the last about 15 plus years I've spent in laboratory sales for about 5 years, and then I worked my way in-house for about 5 years working for a large regional laboratory where I was involved in marketing and operations and product development and product launches and billing and all aspects of pathology, both on the anatomic side and the clinical molecular side for cumulatively up to about 15 years on that. And the reason why I bring that up is not to impress you, but it's really to impress upon you that my background is not scientific. And so the goal here is not to try to debate the merits of PCR testing for infectious disease. As a laboratory, we have come to the conclusion that is a great value add for our laboratory. Our current laboratory that I started and set up does primarily infectious disease testing exclusively. So we have found it to be a very, very beneficial service to both our clinicians and the patients that they serve. And so I just want to point out that not a scientist, and so I'm hoping for those of you that are on the clinical side, how you can work with your product management, product development, sales, and marketing team to look at various menu options, and hopefully this will help you to make a good, solid decision as to whether onboarding this type of testing makes sense for your laboratory. And in general, all of that background I talked about, especially on the lab side, And even as I set up my laboratory about 6 years ago, I set it up really from scratch. So no CLIA, no physical structure, no LLC in place, no website, really starting it from, from my kitchen table. But I took the same approach as I did when I was working for that larger regional laboratory. Is it good science? Will the clinicians use it? Will they understand it? And then, of course, because it is a business side of things, is will I get paid? And so I've kind of modified that a little bit with, assuming that you take a look at it and decide that PCR is good science. How can I work this into my laboratory? And I came across that picture. Some of you may have seen something similar to it on the right, where it talks about the design versus user experience. And there's a lot of tests out there, whether you find them through various outlets like online, social media, medical journals, that look to be great and promising tests as they are designed or how the workflow is developed. But in the practical world of how the clinicians work, maybe it doesn't fit, or it doesn't really add value, or it takes too long and it doesn't help them better serve their patients. And so you always have to keep that in mind in the big picture of, it looks great on a spreadsheet, or the results look very promising in a study. But if you are a commercial laboratory, or you're a laboratory trying to serve physicians and their patients, will they actually benefit from that? I think that if you take it from that perspective and work backwards, it's easier to figure out, to say, hey, this is something that we should adopt with our laboratory. So the first is, can I, or how can I work this into my laboratory? When we're looking at these products and services that, various companies will tell you, we can do this test, or we can offer this service, or we now have this type of platform. Could this new test or service offer good clinical utility, and is it practical for my laboratory? There's going to be a lot of considerations that you need to take into effect in terms of, especially when it comes to, is it practical for my laboratory, in terms of, do you have the space to do it? Not that you can't do it in an existing space or expand your space, but something to consider, the footprint that it requires. Do the personnel I have the capability to understand this type of testing, and the skill set to bring this type of testing on board? If they've been doing primarily like clinical type testing, molecular testing could be a whole new animal for them. That they might need to get trained on as well, or you might need to add personnel that already have that experience with it. And this also can apply to your lab director as well. You might want to have a look into the lab director side of things and see, does your lab director have experience with this as well? Because that will definitely help your onboarding if you have a lab director that has experience with molecular testing. So when we've looked at expanding our test menu, we looked at a number of different platforms. I have a lot of experience with various platforms. In a previous company, we used Luminex and the XTAG platform when we brought on a GI pathogen panel. When I started this company initially, we started out with the BioFire film array, doing just GI and respiratory panels. But as we look to expand the menu, we really wanted to be able to customize panels and also add on antibiotic resistance, especially for some of the testing that we were doing, where we know that antibiotic resistance in general is a growing problem, but it's also a specific problem for some of these testing services that we were looking to offer. So we looked around the various platforms and companies. We settled really with Thermo Fisher, and like I talked about before, with that picture with the sidewalk, is it allowed us to really work backwards from that clinician in terms of being able to build a customized pathogen panels. We could talk to the clinicians we served in terms of what is it that they're looking for. We're able to talk to infectious disease doctors to help us determine if there was anything else we should be looking at, as well as looking at what assays were available to also build in an antibiotic resistance marker for those tests. As well as having some off-the-shelf options. While we have built some, majority of the testing that we do is built off of these custom panels, we do also use some of their off-the-shelf options, especially with the respiratory testing. It's a great panel, and you don't have to go through the customization process. You can just order right from their catalog, which is very, very useful for that type of testing. And also, and the last two kind of go together in terms of being able to accommodate various workflows, also multiple equipment options. And that really has to come down to is how you're going to grow the business, which I'll talk about in the next slide or so. And again, the biggest thing with Thermo Fisher for us was the scalability of those platforms. And when we go, like I said, when we go to the next slide, you'll see how we were able to incorporate various combinations as we grew, sometimes as expected, but with COVID also, how we were able to accommodate the growth unexpectedly with higher volumes because of the options that they allow for. And so this, I think, is a great slide just because, a picture tells 1,000 words. This is really the real life story of how Wise Diagnostic Systems started and grew over the past 3 or 4 years, incorporating the various systems that Thermo Fisher has to offer. And so I'll start with this right here in 2013 and how we went out of the gates here. And again, we weren't sure. We were kind of designed to be a smaller laboratory doing larger panels, so kind of a lower throughput. And again, starting out, we weren't sure. We knew we had a couple of customers lined up who were anxious to start with us. But again, we wanted to make sure that we were being financially sound with the resources that we had. And so we went out the gates with the Duo Prime, which has the ability to do about 12 extractions at a time, and a Quant Studio. And then we use the TAC card system, which is a really elegant system that has these cards that you're able to do your positive and negative control and still build in a custom panel on there. So we had the ability to run about 6 tests per card that included both the pathogens that we were going to detect, and also we simultaneously ran the antibiotic resistance markers at the same time. Even though sometimes when it's negative, obviously for compliance, if the pathogens detected or there were no pathogens detected, we wouldn't report or bill the antibiotic resistance markers because there's no need to run it at that point. But it allowed us to go out the gates and grow the business in a manageable format. And then since we knew at a certain point, if we were running 2 cards, 3 cards per day, it would essentially allow us to then move up to the next platform. So we were growing with that. And we actually started up at the end of 2019. We had really expected an increase in the beginning of 2020. But of course, as everyone's aware, the world kind of shut down in 2020 as COVID reared its ugly head. And with the platforms that we had, we were fortunate in that Thermo Fisher was ahead of the game, already looking to build the kits available to test for COVID. And the customers that we currently had were clamoring for the COVID testing because the state governments were telling them they had to test patients and employees and things of that sort. And so we found ourselves going from being a laboratory that ran a smaller amount of tests that were larger panels to a laboratory that was doing dozens, if not hundreds of tests a day for just the one COVID test. And so with the help of Thermo Fisher, we actually added a second Quant Studio. We adopted the 96-well format for COVID testing, and we added a Kingfisher as well. So we were still able to do our infectious disease panels. A lot of that kind of volumes went down just because everything was so heavily focused across healthcare on COVID treatment and COVID testing. But it allowed us to both continue to do the infectious disease testing on the non-COVID infectious disease testing on the TAC card system, as well as test for the higher volume COVID tests or samples that were coming through the door as well. And then as the pandemic started to ease and COVID became a little bit less rampant throughout the country and throughout the state of Texas, where we primarily operate, we were able to continue to do the COVID testing that was still coming through the door. We were able to grow the non-COVID infectious disease testing using the TAC card system. And for a number of our panels, the volume has continued to uptick. And so we actually added a second Kingfisher 96 platform. The tests that we had been running 2 to 3 cards a day. Now it made sense to move to the open array format. And also we have lower volumes of the respiratory panel. So we actually continue to do the respiratory off the shelf TAC card for that. So as we stand right now, we've kind of transitioned to this system, which gives us tremendous flexibility in terms of what we can test on what machines. It gives us scalability. We still have a tremendous amount of room for growth in there. And as the systems grow, we can continue. If we had an uptick in respiratory testing, we could shift that from the TAC cards to an open array format and be able to continue to keep up with that. And so where we stand right now, we feel really good about our ability to grow our non-COVID infectious disease testing, as well as accommodate if there happens to be any surges in the COVID testing, and just continue to grow our business through the next 2 to 3 years at least, with just the platforms we have existing here in the lab today. Here's some other considerations, and I think that it's something to really understand because of the flexibility the Quant Studio gives you. It does give you the ability to update what you've customized for your panel or add new potential infectious disease panels. But there's just some things to think about. Let me just bring these up. So one is addressing new pathogens. We see them all the time. You see them on LinkedIn. You see them in the CDC updates and emails that they send out. They're continually tracking new pathogens that may pop up on there. And so I just want to kind of address this because it really gets exciting when you think that your system has the ability to, as long as Thermo Fisher has the assay, to bring it on board. And it potentially just may or may not make sense. It's a matter of just sitting and kind of taking a look at how does it fit into your overall testing strategy, what you do as a laboratory, the type of clinicians that you serve, et cetera. And so, for example, you have these top 2 diagrams at the top. And this was really, I think, one to kind of keep an eye on. And that was the monkeypox. For a while, it kind of popped up, and everybody was talking about this increase in the monkeypox with it. And Thermo Fisher had it. But for us, we took a look at it, and the markets that we served were probably not going to be affected by monkeypox. And so while it was an option, we decided to at least sit and wait to see how it evolved and how it developed. We didn't jump right into trying to bring it on board because it was the newest thing. Now, that being said, the diagram here on the top right, and I really kind of focus on the graphs of it, something that we see, especially here in the state of Texas, we started seeing these CDC warnings about Candida auris rearing its ugly head. And what we watched in that is over time, it has grown and grown and grown to be a more prominent pathogen, especially in the markets we serve, especially here in Texas. And so as we were looking to grow our wound business, we took a look at this and said, hey, we were doing more wound specimens. It might make sense to, the next time we go to place an order, to update our panel to include the Candida auris, especially because of, again, the markets that we serve. So we did, in fact, bring on board the Candida auris in order to, again, add some value to the panel that was that clinicians were already using for wound care. And so it's just something to consider as you look to build your customized panels or bring on a pathogen potentially by itself. Another thing to think about is the sensitivity, the culture and sensitivity. I'm talking about that sensitivity side of it versus the antibiotic resistance markers. One thing that sometimes comes up in discussions is that when a culture and sensitivity is performed, that specific specimen is placed in there and the various antibiotics, and you're finding that the sensitivity to that specific pathogen that was grown during the culture. So a little bit different than how the antibiotic resistance markers are tested. You're essentially taking a look at that specimen in its entirety, and then you're looking for detections. So you might see a detection for E. coli in let's say a UTI sample, and in that same sample there might be a detection for a amoxicillin resistance marker on there. Keep in mind that with this testing, it does not mean that that antibiotic resistance marker for amoxicillin is specifically tied to that pathogen detected of E. coli. So that's something to keep in mind. However, the argument that I have had with, or discussion I have had with clinicians, is that in general, if you're looking at that test and saying, I see E. coli tested, I see that there's a resistance to a certain antibiotic, even though they might not be tied together, if I make that leap and say it could be tied to that marker and use a different antibiotic or treatment strategy instead, you still achieve the overall goal of having treat the patient successfully the first time, and also helping to keep in line with antibiotic stewardship. So you still meet the end goal, even if there is a chance that that amoxicillin potentially could have treated that E. coli. So that's just something to keep in mind as you're doing the testing, and especially if you have clinicians that are new to the PCR results, if they have these type of questions. The other thing to consider, and this can actually be a more technical one, is that if you have not already been dealing with molecular pathology, take a look at your LIS systems to make sure that they offer that type of testing, or that they have the ability to build in those types of testing for you. My experience has been that when bringing on a product in general, or a new test menu item, or a service, or even just making a change, that the lab staff, the clinical lab staff out there, is very, very good at doing their validations in a very, very succinct timeline on that. Typically, the outlier is going to be on the technical side, because they have to more or less wait for the clinical side to get done to then be able to test and apply those changes or those additions, or if you're doing it from scratch, those panels, to test them in a test environment before you actually go live with it. So that's just something to keep in mind, is that you're talking to your IT folks early on, so that you don't get done with it and say, wow, we're great, we're ready to go with these panels, only to find out that there's really nothing ready on the technical side, which means, you can't really report anything out until you've completed that. A couple other considerations, and this is something that we came across too, is that when we went from the BioFire film array, was considered a moderate complexity, to the Thermo Fisher platform, was considered high complexity. One of the things that we encountered was, because it required us to purchase potentially infectious materials from various vendors, we had to upgrade our BSL level for that to accommodate that, which was fine. It's just something to keep in mind, because again, that's cabinets, it's equipment, it's shelf space, it's things that takes up a larger footprint that you'll have to kind of consider. Otherwise, you can't get in the validation material that you need to actually go through the clinical side of the validation. And then the last, as I mentioned before, just the space available and the testing personnel. The second component, and I think it's really something you should dig into on the front end, is will they use it? And it's to take a look at how your clinicians are currently doing that type of testing, to find out how to make it comparable, or hopefully easier, so that they actually use it. Because in the fast-paced world that our clinicians operate in, if there's a very long learning curve, or it takes a number of extra clicks or steps, either on the front end or on the back end, it's very difficult to get somebody to change from what they're currently doing, because in their mind, it's been good enough for X number of years. So the idea is, how do I make this as easy to use as possible, and demonstrate that either on the front end by making it easier for typically it's not going to be your doctor that is actually ordering the test. It's going to be your MAs or your nurses, and the doctor will just see that patient, say, hey, I want to order this test. And so it's the staff that really has to do a large amount of the legwork. And so the idea here is to demonstrate to them how easy it is to use and how to make it save them time, especially on the back end. And I think that's where the value of this test comes in. And so because it's primarily the staff, not necessarily the physician, it's to make ordering simple. How do we make it? For us, we use one tube, we use one swab. It does not have to be refrigerated. We make the requisition largely, stickers and staples, right? They can check boxes, they can add a patient information sticker on there, they can staple demographic information to it as well. Make the collection easy for the patient or the staff if they have to swab. And then on the back end, we got to show them how the reporting makes it so much easier. For example, as opposed to in the days of how we traditionally get culture and sensitivity, you could be ordering a lab for, let's say, stool study, and the patient goes to a draw station and they get 3, 6, 9, 12 different jars for the ova and parasite, for the bacteria. They'll get a number of jars for 2 or 3 different types of tests. Some have to be refrigerated, some have to be sent in with a certain amount of specimen in it. It makes it very hard for the staff and the patient to collect it easily, right? So the doctor's waiting on that result to make a decision. The staff has to keep up with tying 2 or 3 different reports to one lab order, whereas in this type of testing, it's all in one. And typically, if you do it the way that we do it with using an overnight service, is that if they collect a specimen today, we'll get it in the lab tomorrow and report it out that same day. And so that way, from the staff's perspective, they say, I take this sample or I get this sample from the patient today. I really only have to worry about it tomorrow, and I should have the result in there. And that way you can also say, hey, the value of the PCR is you might be seeing more detections, but it's not because you're getting, we're over detecting. It's because the PCR tends to be more, have the ability to make more detections, which means you're going to have a better chance of treating your patient the first time. And again, one report. So in the event that they have a detection and there's an antibiotic resistance marker on there, they're going to have that answer all at once with the complete picture, so that that physician can make the best choice possible as quickly as possible, especially when it comes to infectious disease testing. Waiting for 2, 3, 4 days for a pathogen, and then maybe a day or 2 after that for sensitivity to figure out which antibiotic to use. Typically, what our experience has been hearing from physicians is that they will order the tests, but then based on symptoms, they may prescribe something empirically. Well, if they say, hey, you just got to wait till tomorrow to get that result, one that helps to foster antibiotic stewardship by just waiting that 1 extra day to give them the right med the first time. And it saves the patient from maybe getting on an antibiotic that isn't going to work, that then has spent the money on it, that then has to get another prescription based on the one that could work. And this is just something to keep in the back of your mind, is that this is actually a quote from my operations manager, that when it comes to the patient or staff side of a collection kit and a lab order and a test, is easier is better than better. And by that I mean that you have to compromise on maybe the number of tests that you can offer because there's too many jars or too many swabs required. If you have to compromise on something like that, but to make it so simple that maybe it's not the all-encompassing everything test that your clinician wants, it's got the 90% solution and is so simple that they're getting results back and they're able to treat those patients as quickly as possible. I know that many are discussions with, especially the gastroenterologists, if you ask them about stool studies, and you ask them, well, if you order a stool study, how often are you getting that result back? And generally speaking, they say it's a coin flip. Whether it's because of the patient maybe starts to feel better, or in more cases, it's just too difficult to collect the stool specimen, or they don't collect it correctly, it's not refrigerated, and it's rejected at the laboratory and just kind of is lost somewhere in the system. And finally, will I get paid? And this is a discussion I think is sometimes very, very important on the lab side of things, because, you know, as a basically a sales and marketing guy for a long time, it's always nice to offer something that seems very, very cutting edge on it. But what you don't want to do is adopt something where you haven't done your homework on the reimbursement side of things. And you get yourself where you've built this nice test, but as a former CEO I used to work for said, it's a going out of business strategy, where you find out after the fact that you've got a number of tests in the door and you can't get paid on it. And it's a little bit difficult too, because you have to do a lot of front end work to kind of call your payers, review the CMS fee schedule. It does take a lot of research on it just to feel good about what you expect to get paid on it. And does it exceed what your cost will be for it? Not just for putting it on the platform, but potentially the additional rent that goes along with a larger space if you have to take on an equipment loan, and those type of factors. So this is what I just kind of put together as kind of simple lab business basics. One is, and I think this is the most important thing that I've seen a number of labs, especially in the last 5 or 6 years, as I started Wise Diagnostic Systems, is don't chase dollars. A number of folks and colleagues that I've worked for will say, and it's all anecdotal, oh, there's this new test and it's getting reimbursed at X, and everyone's excited about it and they want to go for that test. And when people talk like that, the first thing that comes to my mind is, that's great, but I don't really understand a whole lot about the test and how useful is it? Because what you don't want to do is just start building a test menu based off of what you think revenue is going to be, because that's not really a business plan. That's not really trying to serve a market. That's really just trying, like, how do I generate revenue? And the difficult part of the equation is getting your physicians to adopt it and use it, like I had said in the previous slide. So going for the highest dollar lab test really, to me, doesn't make sense. Getting good lab tests that your physicians say they think brings value and clinical utility and helps with patient outcomes on those that can be profitable to help your lab grow and your business grow. To me, those are the ideal tests to go for. And then with that, you're looking to, with those platforms or things that your existing customer base, can you add tests? Can you add services that help you to expand either off that market or potentially go into a new market? So just something to think about. And like I said, confirm the reimbursement upfront. And then there's also obviously other costs to think about. And again, I've kind of gone into this a little bit already. And again, I think that this third point is the most important when it comes to looking at a new test or service, is base the decision on data, not on anecdotes, because I think that we all tend to gravitate towards the ideal situation or to be optimists. And that's not a bad thing with it. But like that first picture on there where you've got the nice, pretty sidewalk, but no one uses it, you really want to look at the nitty gritty. So you want to first try to, as best you can, confirm reimbursement up front. Medicare is sometimes the easiest. You can just Google CMS clinical lab fee schedule, and it'll actually allow you to download a spreadsheet that will tell you what those rates are for the various CPT codes are. However, that's really only part of the story. You really want to dig into your particular MAC and see if there's any LCDs that are in place that will say, yes, you can test this, but we limit what CPTs you can bill for. There's a maximum amount of modifiers that you can use on these particular tests. Or there's other requirements that you need to have in place for you to get paid on these particular tests. So you really want to dig into those LCDs to look at that. And those are typically all available on the CMS website as well. The other thing for your major payers, Blue Cross Blue Shield, United, Cigna, and Aetna, you can take a look at your contracts, especially if you have not delved into the world of molecular before. my experience has been there are some organizations or facilities that base their percentage they think they're going to get reimbursed on a code, especially if you have not delved into clinical before and you're looking at anatomical rates. They don't necessarily translate. And really the best thing to do is to just reach out and make some calls to your payers. They should at least be able to give you a better insight as to how billing those claims will be affected. That's what I've done every single time, and I feel like you get at least a ballpark answer with your major payers. You don't have to dig into every single payer. These major payers will kind of give you the snapshot with it, because some of the smaller payers, they may do better for you, they may do worse for you. But those in most cases will be a smaller percentage and really won't impact the overall average you expect to collect on it too much. The other costs to consider, and these are kind of those paper cuts where you're not talking about the reagents or the supplies or things like that, is some of the things that can add up. And one is the collection kits. You want to have a very easy to use collection kit, has enough room for all the supplies on it, especially if you're handing it to a patient who has to do an at-home collection or go to a restroom inside your facility to try and collect a specimen. Those can be done inexpensively, through companies like Uline, and just get some basic supplies and kind of put them together yourself. Or you can use some of these larger companies that have the ability to build the kits for you, and you can contact them and they'll usually walk you through what you might need or what supplies you might need for that. They're very, very good at they've done this type of things for molecular testing for a long time. So they can actually be a great asset to help you build a very, very good kit that can be as cost effective as possible. And then the other piece of that, especially if we're talking about infectious disease and the speed at which we can deliver results, is overnight shipping. Sometimes that can be expensive, especially if you're new to one of these overnight carriers like a FedEx or a UPS, in terms of what they will charge you per package. One way that you can kind of help offset that, especially in the beginning, if you're trying to build your package count as you're growing the business, some of these companies that offer to help build kits, they also will offer to take on the shipping for you. So you can just say, hey, client X needs 10 kit, client Y needs 20 kits. They will actually handle the shipping for you as well. The advantage to that is, even though they're kind of taking that somewhat away for you, so you don't have your personal hands on that piece of the logistics pie, is that they're actually getting you the better rates that they get, because they're shipping out thousands upon thousands of packages through these carriers every single day. So you'll get some of the best rates in the nation by working with them. And as you grow and you grow, you'll see that shipping tends to be one of those costs that goes up just as your volume goes up, right? Because just about every package that comes in, or every specimen that comes in, means there's a FedEx or UPS cost or overnight cost associated with it. And the other piece I want to touch on a little bit, because I think this is kind of a hidden cost, and it's also a great value add in terms of customer service, is specimen rejection. And that's to minimize it. And that's kind of the broad stroke, minimize it. How do you do that? One is, again, this goes back to another slide, is go off of data, not anecdotes, and take a look at, we track every single specimen rejection by testing type. And also we include on the notes with it what our common specimen rejections were. And they could be anything from, sometimes the fluid is poured out of the transport media. Sometimes the swab is not left in the tube. We look at all those things and we try to say, what are our most common problems that we can either address with clients or address within the lab itself. One of the items, I'll give you just an anecdote on this, is we had a lot of issues with either the swabs not staying in the tube or not being in the tube when we received them, or we'd go to open the tube and they would kind of spring out. It was because the tube was actually slightly smaller than the break point on the swab. And so folks had to kind of bend it over and get it in there, or they said, well, it must not go in here because it doesn't fit. And so we were able to talk to the clinical side folks and say, hey, I know it changes some things when your tube is a little bit longer, but can we up the size of it so that the break point allows the tube just to drop and stay in? And so that allowed us to improve it on our side of things. The other thing we were able to do is, one of the more common issues we had is people wouldn't really know maybe that there was fluid in there, because you can't really see that there's a whole lot in there, is that would spill out, or they would pour it out, not understanding that it should stay in there. And we actually added like a big highlighted, do not pour out liquid. Liquid must remain with the tube, as a big sticker when you open the kit. So that actually has reduced it somewhat on the patient collection side of things. And so I don't think there's ever going to be a chance for you to completely eliminate the rejections that you have to do. I think that people will sometimes incredibly surprise you with how creative they get with doing things the wrong way. But when you track it and you especially follow up with it, it ends up being a cost savings to you, because you don't have to get a recollect. You don't have to ship an extra kit out to replace it, those type of things. Also, in larger laboratories, my experience is if there's a specimen rejection inside the lab, in many ways, it just dies on the vine, and nobody really knows about it until the physician has a patient back in for a follow-up, and they say, where's my result? And then everybody's scrambling for it, only to find out it was rejected somewhere along the way. We try to reach out proactively to say, hey, we understand this was rejected. This is why. There might be a coaching opportunity with the staff. And then also they understand too, in their mind, they say, this makes it easy because all I have to do is send it out. And if there's a problem with it, the lab is taking care of it. It's just customer service 101. And so that is the conclusion of the presentation. Again, thank you to Thermo Fisher for inviting me to give this presentation. And I think we have some time for questions.
Thank you, Scott Brady, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. All right, let's get started. The first question we have here is, in your experience, why would a reference lab use TAC cards to process their infectious disease samples?
I think the TAC card system is a great option if you have a test that is lower throughput, or if you're getting started and want to keep your testing costs as cost effective as possible. So that if you, for example, we have a test that we continue to run the TAC card system because it is only ordered a few times per week or so, and allows us to continue to offer that test and keep our costs down.
Great. Thank you. Now, Scott, you are developing your own custom panels. How do you determine the pathogens or markers that you add to your panels, and why do you include antibiotic resistance markers?
What we like about the Thermo Fisher platform is the ability to do the customization, because we're able to work backwards. We can talk to the clinicians that we serve, as well as the experts in infectious disease, and actually build pathogen panels that make sense for the test that we're going to offer and the markets that we're going to serve. And the addition of the antibiotic resistance markers allows us to give the clinician the complete picture, allow them to pick the right medication the first time, and to foster antibiotic stewardship.
Great, thank you, Scott. You are very passionate about your assay development approach. What are the benefits to laboratories like yours who are adopting this approach?
I think the primary advantage is that we're able to look at the various markets in healthcare and the clinicians that we serve and the patients that they're trying to take care of, and work backwards to build not just a test, but a service around that test that allows the patient to get a better answer, the physician to get a more clearer picture a lot faster, that ultimately, for all of us, ends up with better patient outcomes, which is what we're all trying to achieve.
Great. We're getting some great questions in from our audience here, so we have time for a few more. This next question asks, when developing panels, you need to consider how to get reimbursed. How much does reimbursement from various payers impact the development of your panels?
Well, I first think it's important to look and see, is the test useful and have clinical utility? And I think you start there to see, is it something that could benefit a market that you're already in or that you're looking to jump into. But I do think it's important to look at the reimbursement side of it very thoroughly to make sure that you can actually sustain this offering. Because the last thing you want to do is to have to modify, trim down, or completely eliminate a test offering because the economics don't work. There's just no way around that, unfortunately, in today's environment.
Great. Now, the analytical and validation testing feel like a long and daunting process. How can labs shrink the amount of time or resource that it takes to conduct analytical and clinical validation?
I think there's a few options here. And again, having not had a lot of experience with the validation process myself until we did it with the Thermo Fisher platforms, where I had to be a little bit more involved, at least from the oversight, not with the actual day-to-day process of it. But I think one is, as we talked about before, in terms of how can I prepare for this as a laboratory, is making sure you have the right staff on board, whether it's your laboratory medical director or personnel that have experience with the validation. That's going to be primary. Also, Thermo Fisher themselves have assets available to help you with the process. And there's also a number of consulting agencies out there too that can help to fill in with any gaps you might have in that process. And there are some out there too that can help you from soup to nuts, to go from helping you choose your panel to everything you need to have an inspection-ready test developed.
Thank you, Scott. There are many decisions that lab needs to make when they move from microbiology to molecular techniques. What are some of the key challenges a lab may encounter, and how did you overcome these challenges?
I think for us is more on the testing side of it. We were not a microbiology laboratory to start with, but I think that it's about establishing a comfort level with a new type of testing. People are very familiar with PCR as a concept now because of COVID and the pandemic. But I think what you're trying to do is to show folks that you're not trying to say, oh, this is absolutely better than culture and sensitivity, and you should absolutely do away with it. We're not trying to call anybody's baby ugly here. What we're trying to say is there's a new format here with PCR testing that potentially can have more detections, offer simultaneously antibiotic resistance that could, in some cases, be a better option or a complementary option for your patients.
Thank you. Now it looks like we have time for one more question, so we'll wrap up after this one. Scott, how does a lab source samples and approach potential sample providers?
I think if you're looking for a couple things, we're talking about the validation side of things. I think that there are a number of options out there, from acquiring validation material to get your test up and running. There's a lot more collaboration now than there used to be pre-pandemic between labs. It used to be very, very competitive. But in terms of sharing samples for validation, that happens all the time now. We share with various laboratories as well, and laboratories have shared with us to help each other along, especially during the pandemic when there were supply chain issues and things like that. In terms of actually getting testing specimens in the door, I think that is really about having a sales team or a focus team out there who is talking to the providers who could best benefit from your testing services, and the ones who actually understand the value of PCR testing, and trying to work with those physicians first. Some people call them early adopters. If you have someone that is really only wants to do culture and sensitivity and doesn't believe in the PCR testing, at least not yet, they maybe don't have a comfort with it just yet, then I would say work with them later, but focus on the people who understand what it is you're doing and how it can benefit their patients and their practice, and go for those folks as well, because those folks will actually see the most amount of benefit. And if you have a multi-physician practice, you might have a wide array of folks that are both early adopters and believers in the PCR systems, and some who are just not comfortable with it. And if you have some folks that are using it, and those other physicians start to see how quick we get the reports out, the full picture in one report, some of those folks who are uncomfortable with it may quickly become comfortable with it.
Great. Well, thank you again, Scott. Do you have any final comments for our audience before we go?
I think when you're looking at this is from the folks who are on the clinical side of it is to work with your sales and marketing and product development team. If the two components of that, both the clinical side and the non-clinical side, can work together to see how they can best serve a clinician or their patient for better outcomes, I think that if you're all willing to kind of work together and compromise and build a very, very good test itself and a good service around it, that's the way to be the most successful in what we do in the commercial lab business.
Wonderful. Well, Scott, I want to thank you again for your time today and your important research. And before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, Thermo Fisher Scientific, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until September 7th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, goodbye.